Right. So what I want to talk about today is how you go from mutations to effects. And you guys made mutations in your proteins for the warm-up. And when we get to DNA, we'll talk in detail about what they really are, what a mutation really is. For the moment, we'll say what I'm going to say right here, which is that it turns out that mutations are changes in genes. And they produce new alleles. So if you remember way back in the APO2-1 lab, you guys made mutant versions of your protein that had a different, of that organism that had a different color. And you looked and saw there were changes to the DNA sequence. Well, that's the deal. Right? Mutations are actually changes to DNA sequence, changes to genes. We'll talk more about how that works a little bit later in the course. For today, what's important is that since genes encode proteins, therefore, mutations cause changes in the amino acid sequence. So, to be really clear, therefore, or that would also be called the primary structure. Okay. Therefore, the proteins are made wrong. That is a wrong amino acid at a particular place. And I want to be clear about the iClicker question. It is what you're basically doing is you're ordering. Let me write that more neatly. Ordering a pepperoni pizza instead of mushroom, and you're not converting mushrooms on the pizza to pepperoni, which is a sort of weird way to put it, but let me explain to you what I mean. So, in the sickle cell question, the choice was between the protein's basically made right, and then the amino acids get changed after, and that's what the mutation does. Or the mutation is the protein is made wrong from the very beginning. Right? A was made wrong from the very beginning. B was made right and altered. And the answer is A. It's made wrong from the very beginning. When you change, the gene is like your pizza order. So your standing pizza has, for all these years, been mushroom pizza, right? So that's what the pizza delivery guy is going to take to your house is mushroom pizza. The mutation alters it. So on the phone, when you say, I want a mushroom pizza, it gets garbled, and the order that comes through is pepperoni. And so he brings you a pepperoni pizza. The order, the gene says, make this protein with a different amino acid, with the wrong amino acid. So the protein is made wrong from the beginning. It is not as though you change the order so the guy in the pizza truck somehow transmogrifies mushrooms into pepperoni, or in the case, you transmogrify serine into glycine. Right? And the thing is, the reason why I make a fuss about that is the terminology is really easy to goof up. People will say as a shorthand, this mutation turns serine into glycine. Right? So it sounds like, yes, it transmogrifies serine into glycine. And the answer is no. That's shorthand. It changes the order, the specifications. And so instead of asking for serine at that spot, you now ask for glycine. So the protein is made wrong from the very start. So when you change your amino acids in your proteins, you are simulating not editing the protein, but actually a new gene that would express your new sequence. Okay? And so that's actually a really very important thing. That's why I asked that question, and many people get it wrong. But the idea is that the proteins are made wrong from the start because the orders, the command of what to make, the instructions, have been altered. Questions about that distinction, because it's a big one. Yeah? So the question is, if they're made right from the beginning and they change in the end, they never change. That is, a protein, once it's made, its amino acid sequence does not change. Once it's been constructed, once the amino acids have been glued together, they cannot change. Right, that's another way to look at it as well. Okay, yeah? Yes, so instead of saying turning something into something else, you would say encodes it differently. So the better, rather than saying the mutation changes glycine to alanine, what you say is the mutated gene encodes glycine instead of alanine would be like the most best way to say something like that. That's good. All right. So I want to show one example of the warm-up. Pages, next page. So here is one. Oh, phooey, made it the wrong size. Well, there's this nifty little blob protein that somebody made, and what they said was this. It's a long bit of text. The key parts are the mutations that did not alter my protein shape were when I replaced lysine for a different amino acid that side chain can make hydrogen bonds. Right? The idea is it's similar to lysine. If you scooch over and see if you could, if I scooch over, I guess, you see that he's got a lysine sticking on the outside. It's nice and hydrophilic. It's out on the surface where you want. You put something else hydrophilic there, it's totally fine. He or she said the mutations that messed it up were when you replaced the tyrosine in the middle with something more hydrophilic or more hydrophobic. So you changed the hydrophobicness of that hydrophobic core, and it disrupted it. And many people said similar sorts of things. That was just sort of people's general experience. You can generalize that experience to be basically, so in general, if the new amino acid is similar to the old, there should be probably no effect. If, on the other hand, if the new is not similar to the old, there will oftentimes be an effect. All right. But not always. All right. There were people that made what were quite dramatic changes in amino acid properties from things that were pretty hydrophobic to pretty hydrophilic that had no effect on the structure. Because sometimes, depending on the structure of your protein, it actually almost doesn't even matter what amino acid is there. And there are things in real life where basically you just got to have something there and it doesn't matter what it is. And then there's other ones where it's super sensitive. The tiny little differences like leucine and isoleucine, which are almost identical. I can never remember which structure is which. Some mutations that cause the protein to be made with one instead of the other mess up the protein. It's very rare. So this is in general, but there's no ironclad rule here. And also, as you see, I'm being sort of politician level vague, right? Similar. What do I mean by similar, right? Does it mean just to have a positive charge? Well, lysine and arginine both have positive charges. Are they similar? Well, yes, in that they have a positive charge, but they're sort of different shapes. So the answer is, this is approximately true. And so on an exam, what would I ask? I will never ask you to, let me how to say this carefully. I might ask you to explain why a change had a certain effect, and then you could talk about similarity or dissimilarity. I might occasionally ask you to predict what would happen, but only under very narrow circumstances and with sort of limited expectations, because as you've seen, it's quite difficult to predict what a mutation will do. There's some sure bets and some not so sure bets. And so in general, my questions are going to be explain, which is much easier to do, why this mutation had an effect. Occasionally they'll ask you to predict, but I have to treat that gently because, in fact, in the real world, you can't, in many cases, it's very difficult to make a prediction as you observed. And as I said, there's some obvious ones. Changing lysine to phenylalanine is likely to make a difference. Not guaranteed, but pretty likely. Whereas changing isoleucine to leucine is not likely to make much of a difference. Questions about that before we talk about hemoglobin and all that sort of thing? Yeah, Sabrina. So the exam questions are going to be more like, I made such and so mutation, it messed up the protein. Tell me why that makes sense. All right? I might occasionally say, these mutations have these effects. What might you expect if I make this particular change? Okay? But only under sort of controlled circumstances, because again, predicting is quite a bit difficult. Other questions? Okay. So what I want to do now is talk about the real world, not the flat